Before I go any further, I wanted to tell you that my cats might have looked funny when you saw her on the video, but she's very healthy. There's nothing wrong with her. They're just a very odd breed. They're called the Cornish Rex cat. They don't shed, and they are adorable. The one that was in the last video was Gidget, and my other cat here is Fiona. And I've got a third one named Daisy that I think was in a previous video. But don't worry about them. They're healthy. They're fine. So we were talking about arteries and veins, particularly what's the difference between them. And let's look into that a little bit more. Arteries are all of the blood vessels that are carrying the blood away from a ventricle. Now, this is difficult terminology for some people, so let me try to make things clear. I used to say that arteries were blood vessels that are carrying blood away from the heart, but that was confusing because coronary arteries are bringing blood to the muscle of the heart, right? So that was confusing. So now I say that arteries are blood vessels that are carrying blood away from a ventricle, and veins are blood vessels that are bringing blood back towards an atrium, right? And the only place where the colors are backwards is pulmonary. Now that means the pulmonary trunk and the pulmonary artery, but also when you look at blood vessels in the lungs themselves, remember the pulmonary arteries are the blue ones. It just makes sense. Here's what's really important for parts of this lecture, capillaries. Capillaries don't get enough credit in anatomy courses because we don't have names for the different capillary beds. We can't tell you, memorize the name of that capillary bed. Well, there are a couple we can ask you to name, but in general, capillary beds are so small, they're microscopically small, that they don't get names. However, I want you to know this for the exam. Capillaries are the blood vessels that allow for the exchange of substances between the bloodstream and the tissue cells. Right? So that means every time some cells in your muscles are like, ooh, I've got all this waste product, who will take this away? And they put it into the blood, that's going to happen at a capillary. Every time oxygen is going to enter the blood, that's going to be at a capillary. So when I think about arteries and veins and capillaries, I think about them as being like freeways and then city streets. We know that ultimately, the guy delivering the cases of Pepsi to the 7-Eleven, ultimately, the only place they're going to be able to download all of those materials are going to be in the alley behind the 7-Eleven, right? So that is like the capillaries. The capillaries allow people to get out of their car, walk into a store and buy a Pepsi, or someone to deliver Pepsis to the store. That happens at capillaries. The arteries and the veins, they're the freeways. How did the truck get from the warehouse to the store? Well, that's going to be on a freeway. That's going to be arteries and veins. Altogether, arteries, arterioles, veins, venules, and capillaries, those are called blood vessels because they are tubes that carry blood, so blood vessels. And it's going to be useful for you to understand a little bit about the anatomy of blood vessels because you will need to keep being able to identify them when you look at them through the microscope. Let's look here. I got everything kind of tumbled on top of each other. Let's separate things out a little bit. All right. Okay, that's a little better. All right. So when you look at arteries and veins, when they're right next to each other in a class where you're learning the histology of a tissue, it always looks like the artery has got a smaller diameter than the vein does. It actually doesn't. Well, right now it does. You see, veins, they don't have very much blood pressure inside of them. So they are not made to be stretchy. That's not their job. Their job isn't to stretch and snap back. So when the vein in any part of the body, when it's filled up with blood, it'll look like that. And when you take blood out, it just kind of collapses a little bit, okay? And that's what you see here in the vein, right? It's relatively thin in the wall because remember, I told you they could be made out of hefty bags. They don't have much pressure in them. And so it looks like this interior space called the lumen is relatively big compared to the space of the artery. Now, right now, there's no blood pressure inside of that artery, right? That thing probably came out of a dead animal and it's dead now. So when the blood pressure left, then all the pressure inside the artery left and the artery shrunk back down. Like when you have a really stretchy balloon and you take the air out of it and the balloon goes, right? So the lumen of an artery will have a smaller interior diameter when you're looking at it through the microscope. But remember, if you were looking at it in the real living organism, those arteries and veins are usually about the same diameter in any given point. And look at how the whole wall, how much thicker it is for the artery. Why? Because arteries have got pressure in them, and that pressure requires the artery to be made stronger, so it needs more layers of cells. The artery also has got a lot of stretchy stuff. Do you see this interior lining that kind of looks like an old-fashioned phone cord, telephone cord? That is lots of elastin there, and that is part of what makes an artery stretchy. How stretchy are arteries? When you're taking a patient's pulse, what are you feeling? You feel that little bump, bump. What are you feeling? You're feeling the artery going bigger, smaller, bigger, bigger. So when the ventricle puts out its pulse of blood, the artery stretches. When the ventricle regroups for the next beat, the artery snaps back, stretches, snaps back. An artery can do that, a vein can't do that, can't stretch and snap back. So you can see that over here on this side. You can see that we're showing quite a bit of the elastin. We've got this dense layer of elastin in the artery. There's some elastin around the outside of a vein, but yeah, not in comparison. Alrighty. But capillaries are super important, and I do want you to know that the job of capillaries is not just to connect arteries to veins. We don't need something necessarily to do that. Are they between arteries and veins? Yeah, generally, yes. But that's not why they're there. They're there so that the blood can deliver oxygen to the cells and pick up CO2. It can deliver glucose and insulin. It can pick up waste products. It's where everything gets dropped off and picked up again. That's what capillaries are there for. Also, let me orient you to this. I just love this picture from your textbook. You can see that is the diameter of a single red blood vessel. I know that you've been looking through microscopes quite a bit, particularly in 150, and you probably remember looking at blood cells, red blood cells, through the microscope. And the diameter of every one of your capillaries is almost exactly the diameter of a red blood cell, because that's how they were designed in order to most efficiently deliver the oxygen that's attached to the hemoglobin inside of the red blood cell, to deliver that oxygen to cells that are like sitting here. The membrane of one cell needs to rub against the membrane of another cell, and that's why capillaries are so small. Also, the distance between adjacent capillaries is only about 2 to 5 cells thick. So here's a capillary, 1, 2, 3, 4, 5, another capillary. So they're super small and they're super close together. Let's talk about the different types of capillaries that human bodies have. You could say that humans have 3 types of capillaries. In a way, humans actually have 4 types of capillaries. Look, I'm not an anatomist or a histologist, so don't write that down, but 3 specific type of capillaries, but there's kind of a 4th one that we'll talk about too, all right? Let's start with continuous capillaries. Almost all of the capillaries of your body, if I was just going to set all of the different kinds of capillaries on different scales, continuous capillaries by far the most. In a continuous capillary, the simple squamous epithelial cells that make up the structure of a capillary, they touch each other, and you can see that they come together like that. This pale blue, bluish gray stuff, that represents the basement membrane that you should remember that all epithelial tissues, their feet are attached to this glycoprotein goo called basement membrane. So the basement membrane is represented by this gray stuff. And here we've got a simple squamous epithelial cell touching the next, touching the next. There are places where one cell touches the next cell that is an intercellular cleft that is just the tiniest bit leaky, just a little tiny bit leaky. Capillaries are not continuous capillaries. They're not super leaky. They're just a little tiny bit leaky. That little bit of leakiness is not a design flaw. That is a design function. And we will talk about how very important it is for allowing your blood to deliver the stuff it needs to deliver. Then we've got these capillaries called fenestrated capillaries. Fenestrated means it has windows in it. That's what a fenestration is, a window. And these capillaries have got little fenestrations, little holes that make a fenestrated capillary leakier than a continuous capillary. And so we will find fenestrated capillaries where we need really rapid absorption or where we need a greater volume of that process called filtration. Those fenestrations are known as filtration pores, and they will allow very small molecules like water to go across. Glucose can go across, but larger things like albumin or fibrinogen or cells cannot go across. So fenestrated capillaries are leakier, and you will hear me talk about them a lot when we get to the kidneys. Then we've got a type of capillary that's so weird, it's actually just called a sinusoid. A sinusoid, they are found particularly in the liver. And come on, they weren't even trying. I mean, you can call that a capillary if you want, but right on this space that's completely open, there would be liver cells because sinusoids are particularly found in the liver. And these are capillaries because they're carrying blood between veins and other veins, but they are ridiculously leaky. Now, I said kind of, there's a fourth sort of capillary. Where did I put that picture? All right, thought I had that picture. I'll try to get back to it. But inside of your brain, you have got continuous capillaries. But why am I saying it's almost like another type of capillary? Because in your brain, these spaces here that have got those intercellular clefts that are designed to just be a little tiny bit leaky, those areas are covered with the foot processes of the astrocytes, creating a structure, a histology structure called the blood-brain barrier. So yes, there are continuous capillaries in your brain, but they've been modified by an additional layer of cells to create a blood-brain barrier. And so the rules of the delivery of substances that are followed everywhere else in the human body do not apply to most parts of your brain. So capillary exchange. I have tried to make the point that the only place that things get delivered or picked up is going to be across the capillaries. And that is done by capillary exchange, which we'll talk about at our next video.